So hi everyone, my name is Marvin Whiteley. I'm a professor of biological sciences at the Georgia Institute of Technology, and I'm also a co-director of CF Atlanta. And what I'm planning on doing today is giving you a little bit of sense about what my lab is interested in, kind of some things that we've been up to over the last 5 or 6 years, and then what we're planning on maybe in the next 2 or 3 years in the future. I'm also going to give you a sense about what it's like here at CF Atlanta. My lab recently moved from the University of Texas at Austin 3 years ago to Atlanta. And so today I'm just going to start a little bit by telling you why we moved. Why would you move from, especially a Texan? I mean, someone that grew up really close to Austin. I was an undergraduate there. I came back as a professor and was there for 11 years. And it's a great place, really good science, really strong in microbiology and chemistry and engineering, which are a lot of the sort of disciplines that my lab is in. And so we moved here probably for a couple of reasons. I think the two reasons are really the two universities that are in Atlanta that I'm affiliated with, and that's Georgia Tech and Emory. And most of you probably know both these places are really great schools, but they're really unique and distinct in what they're doing and what their real strengths are. And so my lab is currently on campus at Georgia Tech. And as you all know, Georgia Tech's a really great science and engineering school. And this really made us feel at home and really provided us with the infrastructure that we were sort of using in Austin. Particularly, the other real strength of Georgia Tech is computational biology. So my lab, while we're in microbiology lab, and I'm going to tell you a little bit about that in a minute, we do a lot of computational biology. So Georgia Tech is a really nice place for the lab. But Austin's also really good in all of those things, and I would say pretty comparable to Georgia Tech. So probably the real reason that motivated us to move here was the interactions with Emory and the medical school at Emory, but particularly the CF Center, CF Atlanta at Emory. My lab, when I began my journey in science in the late 1990s, and I was a grad student at the University of Iowa, and I was working in a microbiology lab, and specifically I was working on Pseudomonas originosis. So all of you, I'm sure, have heard about Pseudomonas originosis. You know a lot about Pseudomonas. It's this scourge of CF. Some of you may not know it's also really important in a lot of other infections. You see this a lot in ventilator-associated pneumonia. You also see it's the most predominant pathogen in chronic wounds now. And so it's this bacterium that is out in the environment. It infects humans, but only humans who have specific comorbidities. So, for example, cystic fibrosis. For wounds, it's oftentimes diabetes. And you probably have heard of diabetic foot ulcers, where Pseudomonas infects. And so, but in grad school, I wasn't really thinking much about infection. And it was really a great time to be in grad school if you were a microbiologist and a bacteriologist like I was. And I was being trained in bacterial genetics. But it was, like I said, a really great time because what we were starting to understand about bacteria is that bacteria were not these single-celled creatures that we once thought they were. So a lot of microbiologists, and particularly biochemists, thought bacteria were just bags of enzymes. Maybe they could swim around a little bit, but they just did things. They ate things. They made proteins that would make people sick. Out in nature, they broke down, wood, and they could interact with plants and cause plants to grow better. And we knew all these things about them, but really we thought that really what they actually did was mostly at a single cell level. And in the late 1990s, it really became apparent that just couldn't be further from the truth. And actually most bacteria are very social. And when I say that they're social, that means that they can actually tell where they are and tell who's around them, particularly from a microbial perspective. And at the time, Pseudomonas became the bacterium, the real model bacterium that everyone was using to study the sociality. Pseudomonas is really, really social. It can actually detect not only other bacteria that are around it, so if Staphylococcus aureus is around it, it can tell that there's Staphylococcus aureus there. But it can also tell how many of itself are actually around. So Pseudomonas can actually count. And this is something called Quorum Sensing. If you want to know more about it, there's lots of videos on YouTube about it. And Bonnie Bassler gave a great TED talk many years ago on Quorum Sensing if you want to know more background. But basically what it means is that Pseudomonas could actually, as it was growing, it would count its cell numbers and count how many Pseudomonas were around. And you think, well, that sounds pretty cool, but why would we care about it? Particularly from a cystic fibrosis perspective, right? Why would we care if Pseudomonas can count? And the reason why we were interested in it is because Pseudomonas actually had distinct behaviors when it was at low cell numbers, cell densities, and at high cell densities. So again, we're thinking about densities, so we're thinking about cells per unit volume, right? And so when Pseudomonas, it turns out, was at low cell density, it didn't make virulence factors. It didn't make things that make people sick. It didn't make proteins and toxins that made people sick. It was only when it transitioned to this high density phase, it would begin to make these factors that would make you sick. It's actually been, and a lot of people have thought about this as a sort of mobilizing an army. I don't know that it's the best analogy in the world, but I do think that it does provide you with some context. And so when you have an army and it's not very dense, there aren't a lot there. Maybe you don't actually attack, maybe you don't cause problems, right? Particularly, let's say for Pseudomonas. If it were to be at a low density population and start making a lot of toxins, a lot of people have proposed that, well, maybe your body could actually sense this damage it was causing, come in and clear it out because there's not a lot of bacteria. Instead, what Pseudomonas does is it waits until it gets to a large number of bacteria. When it gets to that stage, it can begin to make toxins, and even though the body responds to it, it can't clear it. And so I was spending a lot of time thinking about this, and I was doing a lot of molecular genetics. And so fast forward a few years, when I started my lab, we were still working on this and trying to understand, well, how does it count and what does it mean when it counts? But we were really doing this in the lab, and we were doing this mainly in test tubes. So we were in test tubes and growing bugs just in test tubes. And most of you probably don't know, but most microbiology labs on the planet grow bacteria in, we call it growth media, and it's just media that you can buy from a number of different companies. And what these growth media are actually made of are just extracts of things like soybeans, and some of them are made of extracts of brains, not of human brains, obviously, but animal brains, where you basically extract the nutrients out, you sterilize it, right? And then these have historically been, for 150 years, known to be really growth media. So you can grow the bacteria, and they like to grow in this in the lab. That's usually what we use. And so that's mainly what my lab was working on. And then we really sort of transitioned in and started thinking about cystic fibrosis, because I really wanted to move from the lab into the clinic. I wanted to try to ask how my research could actually inform these infections that are so devastating to people. And we really focused on cystic fibrosis and chronic wounds. And one of the first grants I ever received as a new professor was from the Cystic Fibrosis Foundation. And what they funded us for was something that my lab still works on. And what my lab works on now, and one of the things we've been working on for the past 14 or 15 years, is actually trying to get away from those growth media I was telling you about, these random growth media that have been used for over 100 years to grow Pseudomonas. And really, what we decided we wanted to do is we wanted to try to find a growth medium that actually more represented what was actually happening in the CF lung. And you say, well, why might you want to do this? Well, the reason that you want to do this is because bacteria are these amazing creatures, and Pseudomonas is particularly amazing. Now, I've told you that it can actually interact with and count other bugs that are around it and see what other bacteria are there. But the other thing that it can do is it's very responsive to its environment. What I actually mean by that is Pseudomonas can actually tell what's the pH, what's there to eat, what's the temperature like, what's the viscosity like, is it really viscous, is it not viscous? And all of these things that it senses actually dictates how it behaves. And that behavior impacts what kind of disease that you actually might get. And so obviously one of the things, and a lot of you may be thinking this, well, obviously if you're growing this in soybean extract in the lab, it's probably not going to tell you a lot about, that's probably not very similar to how it grows actually in the CF lung. And that's actually true. And that's sort of what motivated us to actually try to understand, can we come up with a way to grow it in the lab that gives us some insights into what's going on in the CF lung? And so what my lab actually did is we actually went in and this was early 2000s. This was a time when most folks with CF could expectorate. And so we went in, they expectorated out they expectorated sputum out in the clinic. The clinicians actually gave us this sputum. And all my lab did is we went in and we just did chemistry. And all we were asking is what's in it? What's in this sputum? And then we also did some things like measured viscosity and these other physical parameters we thought might be useful. And then it was very simple. We found out what was in it. We ordered all those things from a company, mixed them up in the concentrations that you usually see in sputum from CF, and then grew Pseudomonas in it. And what we found was that we got some clues that the Pseudomonas that was actually growing in this medium really differed from how it actually grew in the soybean extracts and the other media that we actually use generally in the lab. And it seemed like a lot of the way it was behaving in our medium that was mimicking CF actually was sort of similar to what we saw in the human CF lung. The problem was really, how do you test that, right? How do you do this in a way? There are some people that were kind of skeptical about this when we first started doing it. And they said, can the environment really be that important? But I always told them, of course, environment's important, right? I mean, if you were studying lions, let's say, and you were out, and let's say you were in Africa and you were following these lions around and studying them and noticing their behavior, you would probably get a certain set of measurements and get some idea about the natural history and the life history of what actually happens to lions. But let's say that you weren't able to do that and you took lions back out and you went in and you actually put them into a zoo, right? And monitored the behavior. A lot of the behavior that you would actually see there wouldn't tell you anything, probably, and would be very unlike what you actually saw in nature. But maybe if you put them on a preserve, right, maybe somewhere in Georgia with 50 acres, and try to make it a little bit like their native habitat, some of those behaviors might be more reminiscent of what you see in their natural environment. That was what we were trying to do. We were trying to get out of the zoo and move into sort of into an environment that we could control, right, but might be able to tell us something. And so one of the keys was really trying to understand, with lions, it's kind of easy. You can just watch what they do, right, and see how they behave and compare the behaviors. It's a little harder to do in bacteria. And so one of the things that about 5 or 6 years ago that we decided to do, there was a technique that finally became cheap enough that we could actually do it in the lab. It's called transcriptomics. And what it actually does, you don't need to know exactly what it does, but what it does, it allows us to go into bacteria and using molecular biology, figure out what the behaviors are in mass, right? And monitor hundreds of these behaviors simultaneously. And so this actually provided us with the opportunity to really do the experiment we wanted to do, compare our medium to really what it's like to be in a human. And so the problem that we had, and we were currently in Austin, as I said, my lab was in Austin, is, in Austin, like I said, was great and had all the technology, but we really didn't have access to a clinic. And this really spurred our move, as I said, to Atlanta and interacting with CF Atlanta and the clinicians over at Emory. And it's been probably the best move that my lab's ever made, because it allowed us to actually do this final test, right? This big test to actually ask how good our media is compared to, and does it actually mimic what's actually happening in a human. And so working with clinicians, Arlene Sashenko and other folks over at Emory, we actually get samples from the Emory clinic of expectorated sputum and induced sputum, and do the transcriptomics there in these samples from humans and in our medium in the lab, our way of growing Pseudomonas, compared them. And what we found was that we were actually very close, and that a lot of behaviors you were seeing in humans were actually happening in our media, but were not happening in these other media like the soy media I was telling you about. So we had improved significantly. And in fact, it turns out we had improved over a lot of the mouse models, right, that have been used. And as a lot of you may know, mouse and animal models have been really important in infection and trying to identify vaccines and try to understand pathogenesis, the ways microbes make us sick. And so what we've understood now is we have this system that you can grow using a test tube. And you might ask, why would you want a system to use so that you have a test tube? Why would you want to grow bacteria in a test tube? And it's because of the versatility. When you can grow bacteria in test tubes, you can do all kinds of treatments to them. You can add antibiotics to them. One can actually go in and image them and do lots of different treatments. It provides you with experimental versatility to understand something. And so this medium and what we developed and the support from the CF Foundation has been really critical for this. And the last part was obviously this move to Atlanta and the interaction with CF Atlanta. So my lab, we're very happy to be here. We just received a very big grant from the Cystic Fibrosis Foundation to further improve this model that we've actually developed in vitro. And our goal is to actually get this model developed and distribute this model and the way of actually using this growth medium in the lab so that all labs can be actually using this data and hopefully learning more about the CF lung in their work than they were when they were using these other mediums. So I hope that was useful and sort of gave you a sense of what we were working on. And really want to just thank everyone, in Atlanta. I've had a lot of really good interactions, obviously with clinicians over at Emory, but also a lot of folks associated with the Georgia CF chapter and going to a lot of events. There's a great infrastructure here and really appreciate it, and a lot of really passionate people interested in CF. So thanks.